Okay, today we will discuss uh, apolar interactions, and one of the most important apolar interaction that is found to be stabilizing your usually protein ligand complex is in form of pi stacking interaction. Okay, so now these pi sticking interaction are created in your protein ligand system by the virtue of the aromatic rings that are usually present in your protein molecule. Okay, so there are amino acid residue which has got aromatic ring. So you can consider that amino acid residues like phenylalanine, tyrosine, histidine, and tryptophan. Well, all these residues are having some aromatic phenyl ring, and these aromatic phenyl rings can be employed in stabilizing the protein ligand complex if there are any phenyl ring or aromatic ring in your ligand as well. Okay, so let us start out with demonstrating key when is that you would when is that when you would suspect key there is a pi sticking interaction. Okay, so I have a system ready. Okay, just a minute. Okay, this is some paper that I was working on. This paper was all about some chemical molecules that one of my friend at medicinal chemistry has synthesized, and they checked the antiplatelet activity. Okay, so we have identified one of the target molecule, and then after performing molecular docking, we ended up identifying that there might be some apolar interaction in form of pi sticking interactions that can stabilize this molecule in the receptor, protein receptor as such. Okay, so now let us try to visualize how many molecules are there, or how many amino acid residues are there that are in vicinity of your ligand. Okay, so I would label these residue over here. Okay, so here we go. Now what you can see over here is that this is your ligand molecule. Okay, and it contains some aromatic centers, right? So this particular ring can be considered as aromatic, right? And this ring also can be considered as an aromatic. So you can clearly see that this ring from your ligand molecule is readily available for pi stacking interaction with aromatic molecules like tyrosine 105 and tyrosine 109. Right, the phenyl ring from this tyrosine molecule can readily be stacked upon this much portion of your ligand molecule, and it can be effectively stabilized by pi stacking interaction. So is the case with this phenylalanine residue and this aromatic ring. Okay, so in this way what you can do is that if such is the situation, you can suspect ki, okay, there are a lot of aromatic amino acid residues around my ligand, and therefore they can be effectively be engaged by this pi stacking interaction. And it can also go the other way around, because if you have detected that in my protein, if there are a lot of aromatic amino acid residue, then I would kind of design an inhibitor that would contain aromatic side chains as such. Okay, so that these molecules can be effectively localized, and these molecules can be effectively placed in the active site of your protein. Okay, so such kind of structural analysis and such type of design will give you a better idea in designing a proper molecule that would go and inhibit your protein target as such. Okay, then this is how you can detect key. Okay, these are the residues that can, we'll say for example in our case tyrosine 109, tyrosine 105, then tyrosine 259, you have got phenylalanine somewhere. Yes, this phenylalanine 252. Well, these side chains can be effectively targeted for their putative interaction with some ligand if it contains any aromatic ring as such. Okay, so besides that, you might also consider while designing ligand key there is an arginine molecule or there is a lysine molecule. These can also be used if you have some counter charges present in your molecule as such. Okay, so in this way, analyzing the binding site of protein might give you a kind of idea or give you a kind of structural perspective on how the ligand should be. Okay, so now this is the three dimensional way of identifying if some interactions are present in your molecule. So sometime it happens that you need to represent your these three-dimensional finding in a two-dimensional way so that you can represent those in your protein. Okay, so there are online software. One of them is called as PoseView. Okay, PoseView is a software wherein you can detect if your protein ligand complex contain pi stacking interaction or not. Okay, so what you are supposed to do in that software is that you are supposed to load your protein molecule, load your ligand molecule, and then it would give you a two-dimensional depiction around your molecule as such. Okay, so over here as I have suspected, it is showing that this phenyl ring of this tyrosine might get stacked upon this much moiety of your ligand molecule. Okay, phenylalanine from this one, this particular 250 second position can pi stack with the aromatic ring present at this location of your ligand. Okay, so in this way it becomes effective for your presentation as well. Okay, so now I have told you that what are the different ways by which you can first detect the pi stacking interaction if they are present in your protein molecule. Okay, and the next way around is what are the softwares that are available for you to present those depiction in your thesis or in your research paper. Okay, so use these softwares very wisely. It becomes very effective when you write down your thesis or when you try to present something in your seminar or in any conference, or we'll say your thesis presentation as well. Okay, so that was how do we visualize, how do we identify if there are pi stacking interaction which are a type of apolar interaction. Okay, so this is what I have visualized. It was one of the other protein complex molecule that we were working in some time back. Okay, so now another type of apolar interaction that exists in your protein ligand system can be in form of van der Waals forces. Okay, uh, as well as you can say that your hydrophobic interactions can also be included in apolar interactions. Okay, so now this van der Waals forces can arise as a consequence of numerous hydrogen bonding interaction that happens between the protein and ligand molecule. Okay, so what happens is that this hydrogen bonding interaction might enable other atoms in your protein ligand system to come closer to each other, and thus they can be stabilized by your van der Waals interactions as such, or van der Waals, they can be stabilized by van der Waals contacts. Okay, so We are going to deal with visualization. We'll say, for example, 2D visualization of hydrophobic interaction and van der Waal contacts just in a while after some time in this lecture itself. Okay, so now let us move our discussion from apolar interaction to electrostatic interactions. Okay, so there are electrostatic interactions that are present in your protein ligand system or even in your single protein system, and these electrostatic interactions are called as salt bridges when it is, you know, described only in term of protein molecule. Okay, so what are salt bridges? Salt bridges, you know, are produced due to electrostatic interaction. Okay, now how do they differ from your polar interaction? Polar interactions are caused due to uneven shifting of electron density toward one of the electronegative atom. Okay, and hence we say that when a chemical species get polarized, one of the component gains a delta negative charge while another component gains a delta positive charge. Okay, so there is differences in there is polarity in your molecule induced by the electronegativity or differences of electronegativity between the two atoms in your chemical group. In that sense we say that there are polar interaction, and such polar interactions are usually in form of hydrogen bond. But electrostatic interaction differ quite significantly. How? Because, you know, these electrostatic interactions are formed due to charges that are developed in the side chain of your protein. Okay, and these charges can be developed by either loss of electron or gain of electron at physiological condition. Okay, so well, say for example, at physiological condition, your glutamic acid can lose one of the proton at the side chain, and hence we can say that it has attained a glutamate state in physiological condition. Okay, and since it has lost since it has lost that proton, it will gain a negatively charged. The opposite thing can happen in case of basic amino acid residue like lysine. They can, you know, acquire a proton and gain a positive charge. So if in case such oppositely charged side chains come closer to each other in the protein molecule, we say that these two side chains are experiencing electrostatic interaction, and we call such electrostatic interaction in protein as salt bridges. Okay, and you should note that these electrostatic interaction or salt bridges are non-covalent in nature. Okay, but they are very strong. They can keep hold of the two side chain very strongly as compared to your hydrogen bonds or your apolar interaction like pi stacking interaction or van der Waal contact. Okay, so these salt bridges involves direct interaction with opposite charges, and hence they differ from hydrogen bonds in the sense that hydrogen bonds are caused due to polarity of the chemical species that are involved in it. Okay, so you can see over here like lysine can form a salt bridge with glutamic acid. Arginine can form a salt bridge with aspartic acid, and so on and so forth. Okay, so well, now let us discuss how people have tried to classified these salt bridges. Okay, so the first way of classifying the salt bridge was on the basis of where exactly these salt bridges are located in your protein. Okay, so well, say for example, you can have exposed salt bridges like this. Okay, and as well like this. Exposed salt bridges are supposed to be formed when the charges that are provided by those side chains, and the side chains are present on the surface of your protein. We say that that salt bridge is exposed. Okay, then you can have a fully buried salt bridge, as you can see over here. Such a type of salt bridges are formed when both the positively charged and negatively charged side chains are buried deeply inside your protein. You can have half buried salt bridges, okay, as in case of D कहाँ पर है? यहाँ पर है. One of the protein is on surface while another one is in core. Sometimes it also happen, consider over here, that many of positively charged and negatively charged side chain can cluster in one location of your protein, and they can form a complex network of salt bridges, and we call it as complex salt bridge. Okay, so these are different type of salt bridges that can exist in your protein molecule. There is another specific example of salt bridges that can be formed due to positive and negative amino acid residues that can be present in vicinity of each other, since common secondary structural elements like alpha helices, they have, you know, a tendency to become compact uh, as compared to coiled coils, or as compared to your beta sheets. Okay, so these due to presence of oppositely charged amino acid residue in vicinity of each other, and that too on helices, well, that can form another type of salt bridges as such. Okay, so that was the type of salt bridges that one can classify. You can read this paper for more details of what are different type of salt bridges. This is where I have this paper is the source of this information. Okay, so now let us discuss uh, well, you know, what are other electrostatic interactions that can be, you know, a reason for stabilizing the salt bridges, or that can result in destabilization as well. Okay, so Kumar and Nusinov back in 2002 tried to put forth this concept based on what is the distance between the oppositely charged molecules. Okay, so you can call it as salt bridges, but at different length. Okay, well, when we say that salt bridge exists when the two oppositely charged molecules are present in vicinity of at least 4 Armstrong unit. Okay, then we say that there exists salt bridge between two molecules. Okay, but what if there exists a situation in which the oppositely charged side chains are present in distance of 4 to 5 Armstrong, and in such case we say that such two side chains form NO bridges, or something called as nitrogen oxygen bridges. Okay, well, say for example, if the distance between two oppositely charged side chain is greater than 5 Armstrong, we say that there might be a longer range ion pair, or what we call as LIRP. So now, based on whether the two side chains, whether they are in form of salt bridges, or whether they are in form of NO bridge, or whether they are in form of LRIP, or longer range ion pair interaction, well, they can either impart stabilizing effect on your protein, or they can destabilize your protein. Well, so for example, if the two charges are present in quite vicinity of each other, over here you can see that these two charges are present at a distance of only 2.9 Armstrong. In such case, they have got the strongest strongest you know strongest interaction, you can say. Okay, so NO bridge are weaker than salt bridges, and LIRPs are the weakest as compared to salt bridges and NO bridges. Okay, so now what is the effect of the presence of salt bridges or NO bridges or LIRP on the stability of protein? Okay, so these salt bridges are known to provide a very strong stabilizing effect in your protein. Okay, while if the two charged molecules form NO bridge, well, they can be considered as considerably reducing stabilizing effect. They can impart considerably reducing stabilizing effect. While if there are longer range ion pairs, well, these are known to destabilize the protein molecule. Any charge system is usually not very stable. Okay, so it becomes thermodynamically, theoretically, it becomes difficult to call it as a stable molecule if there are unbalanced charges that are present in your system. Okay, now since these LIRP are present at quite large distance, that is greater than 5 angstrom, you can consider that these molecules, can sometime form alone LIRP, and most of the time they won't form that LIRP interaction. Okay, so that is why these are considered as destabilizing interaction in your protein molecules. Okay, so you can consider this paper for more details of what are different type of electrostatic interactions that are present in your protein. Okay, so let us now move on to other interaction. As I have discussed already, that there are sometimes it happens that for activity of certain protein, it is essential that some ions or cofactors are required. Okay, so well, say for example, in your C type family of lectins, they require presence of calcium dipositive ions to recognize their carbohydrate ligand. Okay, so lectins are carbohydrate recognition protein, or what we call it as CRPs. So in order to recognize the carbohydrate substrate, they usually require some calcium ions to be present around. Okay, another example can be your magnesium ions are required for RNA folding. उसको proper RNA folding के लिए it is essential that magnesium ions are required. Okay, then your famous example is that of hemoglobin that requires iron, iron for carrying oxygen from one location to another location. So there are specific family of protein called as zinc metalloproteinases that require zinc ion to be present around. Okay, so many of the protein molecules require certain cofactors that must be present in its vicinity so that it can function properly. Okay, and usually what happens is that once this cofactor comes in its location, or what we can say that cofactor binding site, which is usually present near the catalytic center or near the inhibitory binding site, what happens is that these cofactor molecules are strongly coordinated by certain side chains. Okay, well, say for example, in case of hemoglobin, this iron molecule is strongly coordinated by the imidazole group of histidines that are present in its vicinity or in its surrounding. Okay, so there are specific residues that are usually associated with coordinating with certain ions. Okay, so that is also one of the important interaction that exists in biological world that impart stability for your cofactor molecule in or around your protein's binding site. Okay, so that was all about your coordination interaction. Well, now let us talk about visualization of the active site. Okay, so now how would one define an active site? This is a very important topic that I want to discuss. क्या होता है active site? Yes, what are active site in so far your protein is concerned? Are you guys there?
यस सर।
हाँ,
सर
क्या
जहाँ पर
क्या होता है
एंजाइम
एक्टिव
बाइंड
साइट? कोई बताएगा मुझे?
सर जहाँ पर एंजाइम मतलब बाइंड करके एक्टिविटी दिखाता है।
Okay.
हेलो
Yes, yes, you are
सर।
audible. You are audible.
सर ये मतलब एंजाइम में रहता है एक्टिव साइट, मतलब
Mhm.
लिगेंड एंजाइम के साथ मतलब एक्टिव साइट साइट पे बाइंड करता है।
Okay. Uh, you know, your answer is quite logically correct. Okay. So the thing is, active site is a small place in your protein, you can say, and usually that active sites are present quite deep in the core of your protein usually. Okay. एंड एक्टिव साइट का एक स्पेसिफिक बायोकेमिकल डेफिनेशन है. ओके. वेल से फॉर एग्जांपल, you know we say that when specific, or you can say when some special side chains orient in a specific way in the core of your protein, that forms certain functional chemistry in that protein, right? And that specific arrangement of the active site residues side chain in your protein would enable your protein to perform certain function. Okay. So in order to call a certain location in your protein as an active site, it is necessary that it involves orientation of, say, some specific amino acid residues to create a functional chemistry over there, so that this functional chemistry would enable your protein to do what it is designed for, right? Do you agree with this concept? डू यू अग्री विद दिस कंसेप्ट कि कोई भी अपन एक आपके प्रोटीन में अगर कोई सरफेस होगा एंड देयर माइट बी अ क्रीवाइस, यू कैन नॉट कॉल इट एज एन एक्टिव साइट. एक्टिव साइट अपन उस पॉकेट को कब कहेंगे? when certain specific amino acid residues side chains are oriented in a specific way, so that it would enable that environment to be created, a specific chemical environment to be created in that small pocket, that would enable that protein to perform its function. Then we say that this is the active site of your protein. Okay. मैं ये चीज इसलिए बता रहा हूँ बिकॉज पीपल यूजुअली गेट कंफ्यूज्ड with with active site and binding site. Okay. Active site cannot be predicted. Okay. Why? Because for activity of, say, certain protein, there must be certain amino acid residues that must exist over there. Okay. And from structure you cannot kind of predict where is the active site. Okay. What can be predicted is that what are the binding site, or you can say what where are the pockets in your protein, that can be predicted. Okay. So usually there are pocket detection programs that are present online. Okay. So you can consider this pocket finder is one of the program, qsite finder is one of the online program. So what do this program do is that they would detect how many pockets are there in your protein, and these pockets may not necessarily be the active site pockets. Okay. So you should be very discriminating and clear between what you're dealing with. Pocket detection is a different concept and active site is a different concept, because as a reviewer and as I have seen a lot of people doing this thing. What do that they use, they predict the structure of, say, certain protein, and they use these pocket detection softwares and say that this is the active site of my protein, the deepest pocket might be the active site of my protein. Okay. But biologically it might not be the relevant answer. Okay. So active site is a different thing and pocket detection is a different thing. Okay. So don't get confused in this thing. Okay. So now let us try to as one of you have correctly answered, active site is something where your protein or where your ligand molecule would go and attach. बरोबर है? That is what it is said, or what you can say, whether active site is where your substrate molecule would go and attach. Okay, ligand molecule be, it can, if the protein is allosteric in nature, it can go and bind to somewhere else. Okay, so the place in your protein where your substrate goes and attaches can be considered as an active site. Okay, and you can also consider your active site in another way, is that if your inhibitor is a competitive inhibitor, and the place where the competitive inhibitor goes and binds can also be considered as an active site of your protein. Okay, why? Because competitive inhibitor competes with the substrate for its binding, and therefore your substrate and your competitive inhibitor would go and bind at the same location in your protein molecule, and that would be eventually your active site. Okay, so now there are different ways by which you can determine whether is the where is the active site of my protein if you have been provided with the model of that protein. Okay, well, now I would kind of demonstrate you how you can detect what are the active site, or we'll say for example active site residues, how you can determine that these are the active site residues. Okay, so well, say for example I would show you some protein in complex with its substrate or in complex with its inhibitor. Okay, okay, let us work with this one. Well, this particular model correspond to aldose reductase that is complexed with its inhibitor called as zopolrestat. Okay, so this is your inhibitor zopolrestat, and now active site residues can be the one that might be present in vicinity of this particular inhibitor. Okay, so what you can do is that again you can select the amino acid residues that are present in vicinity of say 3.5 angstrom. Okay, and show them. Okay, so these residues, whatever I have selected, can be called as active site residues. Some of these residues can be called as active site residues. Okay, now what you can do is that you can compute the hydrogen bonds. Okay, and you can display only hydrogen bonds that are formed or that originate from your inhibitor, right? Well, say for example, show only hydrogen bonds that are originated from this inhibitor. So you can now say that this side chain, well, say for example, okay, so you can consider that in case of aldose reductase, the side chain of histidine can play an important role, or side chain of tyrosine 48 can play an important role, or even histidine of tryptophan 111 can play an important role in stabilizing this inhibitor in the active site of your protein. Or if there is a substrate molecule that is co-crystallized with your protein molecule, in this way you can determine, okay, since these residues are in perfect interaction with your inhibitor or protein molecule, they can be they can be involved in its catalytic activity. Okay, so in this way, you can from three-dimensional analysis, structural analysis, you can determine, okay, these might be the amino acid residues that might play an important role in either catalytic activity, or they can play an important role in inhibition as well. So then what you can do is that you can design some mutagenesis experiment wherein you can replace this tryptophan 111 by alanine, or you can replace this histidine 110 by alanine, what we call it as alanine scanning experimentation, and then you can test if the protein retains its functional activity or not. Okay, if it still retains the functional activity even after replacing the side chain of these identified amino acid residue, that means that these amino acid residues might not be the part of the active site. Okay, some another residues can be the part of that active site. Well, this is how experiments can be designed by using this structural information as well. Okay, then how do we represent these active sites in in two-dimensional figures as such? तो उसके लिए एक और सॉफ्टवेयर अवेलेबल है that you can readily use. Well, that software is in form of this Leak Plot Plus. Okay, so of course this is a free software. If you have an academic email id, you can download 1 year license for this Leak Plot Plus. Okay, so well, I will just show you how do we visualize the active site of your protein in 2 dimension, or well, so for example, how can you create a pdf of your active site of a given protein, right? तो इसके लिए we need to go to file, open a pdb file. It is quite simple. I would go to this particular location where my protein file is located. Okay, now it will ask you, there are two inhibitor two hetero groups present over here. This is NADP and another one is your inhibitor. So you can select whether you want to show the interaction between this prosthetic group or this ligand group. So for time being, I would select this ligand group and run the Leak Plot. Okay, here we go. So here is your 2D representation of the active site. Okay, as we have already detected that it can form an effective hydrogen bond with histidine 119, which is shown in this green color, and it also shows the hydrogen bonding distance over here. Okay, so you can also visualize the hydrophobic interactions as well. Okay, you can say that this proline, this tyrosine and this cysteine, they can form hydrophobic interaction with this inhibitor, and so is your tryptophan, phenylalanine and tryptophan 219. You see this eyelashes type symbol that indicates that this much portion of your ligand is present in hydrophobic contact with these amino acid residues. Okay, and these shining, or you can say sun type rays that are present over this ligand molecule, those represent that they are in van der Waal contact. Okay, so this carbon molecule is in van der Waal contact with this tryptophan carbon molecule that is present over here. Well, these represent your van der Waal contact, while these eyelashes type of designs, they are they can be considered as your hydrophobic contacts as such. Okay, or hydrophobic interactions. Then what you can do is that you can convert this particular view and save it as a PDF so that you can later on visualize your interaction in more details. Okay, so this is how you visualize your active site. This is how you depict the active site in your thesis, so that it becomes easier in this way to represent say some structural information in your thesis as well as in your research paper. Okay, so use these tools very effectively, very wisely. That can improve your presentation. Okay, so that was all about your visualization of protein molecules and what are the different components or what are the different forces that might exist in your protein. Well, I have left out with demonstrating how one can determine if there are salt bridges in your protein. Okay, any idea? How can you determine if there exists any salt bridge? Well, it is quite simple. Okay, so well, say for example, what we can do, now you know that salt bridges exist uh, between two oppositely charged groups. Okay, so now you can select from over here ki how many charge group are present. Okay, you can go in this select and then you can select group property and maybe select acidic amino acid residues. Okay, and you can display only acidic amino acid residue, right? So well, say for example, now you can hit this control button on your keyboard, then again select, and again group property, we have selected acidic. Now we would select basic amino acid residues. Okay, so now basic and acidic amino acid residues are selected as well. Now I will show these basic and acidic amino acid residues as well. Okay, so now what you can do is that you can color your side chain by properties, right? Select or you can say color by type. Here we go. Okay, so now it becomes very easier for you to determine if there are salt bridges in your protein. All you need to do is to figure out if do some structural analysis and figure out if there are certain side chains that are present in blue color that are present in well, you can say vicinity of we'll say for example, if the distance between the positively charged side chain and negatively charged side chain is less than 4 Armstrong, then you can say the salt bridge exists. It can be the salt bridge can be present over here in our case. Okay, so now how do we do that? Okay, now let me zoom into this portion. For this, what you can do is that you can use this zoom button, just make a view clear, right? And again zoom over here, right? So it appears that this might be a lysine. So how can we identify what residue is this? Well, you can say there is leucine 14 button over here that will tell you what residue it is. Well, this is NZ atom of your lysine 239, right? This is in blue color, and by the look at this, it is possible that it can be glutamate, right? Well, let us identify this as well. This side chain is sorry, it can be an aspartate. Okay, so this is aspartate side chain and this is glutamate side chain. Okay, and we know that the end of your aspartate molecule would contain acidic group, and at the end of your lysine molecule there would be positively charged group in form of that NH3 or NH2 group. So what you can do is that you can just find out what is the distance using this particular button over here. Okay, let us see what is the distance between this atom and then click the next atom, that is 2.89, and hence you have identified that salt bridge can exist between lysine 239 and aspartate 284. Similarly, histidine side chain is also present over here. Okay, well, let us see what is this atom. Of course, this is nitrogen. Okay, so this nitrogen can be involved in your salt bridge. Okay, so you can check it out if this atom is present in vicinity of the salt bridge. Yes, that lies in the range of 2.62. So this aspartate 282 can form a dual you can say dual salt bridge with these two residues. Okay, and that is how you determine how many salt bridges are there. Okay, so this is how you identify, you visualize the electrostatic interactions as well in your protein molecule. Okay, now this is all I have for you today. Okay, I hope you enjoyed and I hope you have learned how do you visualize active sites, how do you identify active sites, how do you visualize and identify the electrostatic interaction, van der Waal interactions, apolar interactions, so on and so forth. Okay, so I would expect that you would use this information in your in your project model, or you can use this information, or you have learned how to do these things now. So better implement it and use it, I mean translate it somewhere. Okay, so are there any questions? Okay, and when you perform such structural analysis, make sure that what atoms you are dealing with. Okay, it should not happen that this particular atom, if it is present from your side chain with that of aspartate, well, you know that over here this atom might not be the nitrogen. Okay, and your carbon atom cannot be involved in formation of salt bridges. So just identify a proper molecule, then find out a distance, and then try to, you know, try to come to a conclusion ki whether that particular interaction can exist there or not. Okay, so that is what I want to convey. Yes, question time. So if there are not any questions, then maybe I would stop the recording.